Hello everyone, my name is Bobby Ranjan and I come from the Genome Institute of Singapore, where I work in the laboratory of systems biology and data analytics. Today, I'll be presenting DUBStepR, a correlation-based feature selection algorithm for clustering single-cell RNA sequencing data. Conventionally, heterogeneity in scRNA-seq has been represented as unsupervised clusters, wherein each cluster represents a unique cell type or cell state. The steps involved have been well described in this review paper here, of which feature selection, that is, selection of the optimal subset of genes to separate cells into clusters, is an important step. Existing methods for feature selection test genes individually. For example, highly variable genes look at the variance to mean ratio of a gene, and M3 drop looks at the dropout rate. Hehowever, we hypothesized that pairwise correlations between genes may have important information for the purpose of feature selection. On the left, we have a heat map of gene expression of the top 10 markers of each of these 5 cell lines, the dataset obtained from this Nature Methods paper last year. On the right, we constructed a gene-gene correlation heat map of the same genes ordered in the same manner. As one can immediately see, marker genes of the same cell type tend to be strongly correlated with one another, and marker genes of different cell types tend to be anti-correlated with one another. Correlations going from blue, negative, to red, positive. Thus, we looked at the range of correlations, that is, the scale difference between the positive and negative correlations, and hypothesized that that would have some predictive power in feature selection. And indeed, we see that this correlation range score is significantly different between DE genes and non-DE genes, and predicts or correlates well with the log fold change and log Q value of differential expression. Thus, we decided to use this intuition to build DUBStepR. In the first step of DUBStepR, we filter out genes that have a poor correlation range score and only keep genes with a large correlation range. However, we wanted to identify genes with unique patterns of expression so that all cell types are well represented. And for this purpose, we perform stepwise regression. We identify unique patterns of expression in the gene-gene correlation matrix by regressing genes out one at a time that explain the most variance in the gene-gene correlation matrix. Using a scree plot based on the variance explained, we can identify an elbow point for the most representative set of regressed genes. We can then add correlated genes to this elbow point feature set so that different sections of this UMAP are populated. The UMAP visualization here is a visualization of the gene-gene correlation matrix, and hence, as different regions start getting selected, different regions could represent different patterns of expression. Now, most feature selection algorithms stop at this point and allow the user to determine either an FDR threshold or a fixed number of genes that the algorithm would return. However, we thought we could automate this process by developing a metric to optimize so that we can predict the optimal number of features required to cluster the data. For this purpose, we proposed density index. Now, the two UMAPs here, the UMAP on the left showing poor clustering and the UMAP on the right showing good clustering, the main difference between them is the fact that on the right, the local neighborhood of cells is dense relative to the rest of the space. However, on the left, the local neighborhoods of cells is sparse. So we leveraged this and quantified it to develop the density index, which is a measure of local neighborhood density, and use that as a stopping criterion for feature selection. The peak of density index, that is the number of feature genes for which density index is the highest, is selected as the optimal number of feature genes. Now, to compare or to determine whether our strategy worked, we devised a benchmarking strategy that involved 7 data sets with objective ground truths, either cell line or fact sorted data, comparing against 6 existing feature selection methods spanning 4 protocols, so 10x, SmartSeq, DropSeq, and CellSeq. The performance was quantified using 2 metrics. First, silhouette index to measure cluster separation, and second would be the AUROC of marker gene detection. By averaging silhouette index after scaling on multiple datasets, we can see that regardless of the number of feature genes selected, DUBStepR is able to, on average, outperform the existing methods. This is even without using the density index, so allowing the user to pick the number of feature genes. Then, after setting default settings, that is, after giving each method its default FDR or number of genes threshold and using the density index on DUBStepR, we see that the silhouette index for DUBStepR is still higher than the rest. Further, since we performed this, since we developed this method based on the intuition of marker gene detection using pairwise correlations, it's no surprise that DUBStepR significantly outperforms existing methods in detecting marker genes. Finally, the intuition behind gene-gene correlations is not specific to genes. That is, the correlated features could also be extended to peaks in single-cell ataxic data. Indeed, if we compare clustering of single-cell ataxic data using all peaks identified in the data against peaks selected by DUBStepR, we find that DUBStepR is able to delineate the hematopoietic trajectories better on this bone marrow SCS ataxic dataset. To conclude, pairwise feature correlations contain important information, and DUBStepR leverages that to perform feature selection for single-cell data. We propose density index as an optimal stopping criterion for feature selection, and DUBStepR is available at this URL on GitHub. Finally, I'd like to acknowledge my lab, the co-authors and lab members who have helped me out so far, funding from GIS and ISCB, and thank you all.